back in the day, as a trainee in Mike Wigler's lab at Cold Spring Harbor, we were using some of his technology that he developed to look at the genome and you know, look specifically in cancer genomes to find mutations. At that time, what actually turned out to be the most interesting stuff were the mutations we were finding in healthy genomes. In fact, there were large deletions, duplications, strange structural mutations in the genomes of everyone walking around on the planet. And that was a shocker that, you know, we really didn't expect to see that much sort of variation in chromosomal structure as part of our naturally occurring genetic variation. We just didn't expect it. So that kind of was really a, you know, a big, you know, bomb that went off in the genetics field that kind of, you know, highlighted this as an area of genetics that really hadn't been explored. It was pretty obvious to us early on that this kind of an approach was probably the best thing going to look for rare mutations in common disease, because obviously genome-wide association studies were in full swing, microarray technology was in its heyday, and you could use those kinds of approaches to find deletions and duplications. And so this was the right kind of an approach to go after, you know, large populations, look for rare glitches in the genome, and compare patients and healthy people to see if they have, you know, certain rare glitches in patients that you don't see in the general population. Sure enough, and that's what we found. So we first, Mike and I started a project on autism in 2005, I think it began. And within a couple of years, we'd collected enough data on families to see quite clearly that there was a much higher frequency of rare glitches in the genome in kids with autism than compared to their healthy siblings. And it just kind of evolved from there. As it turns out, this is not unique to autism. In fact, you know, the majority of severe brain disorders, neuropsychiatric, neurodevelopmental disorders, seem to have a complex etiology which includes lots of rare mutations. There's just something about when you introduce a severe mutation into the genome affecting some brain-expressed gene, what happens is you have pretty significant effects on neurodevelopment. I mean, it can manifest as, you know, severe social impairment and learning disability in a child, or it can manifest later in late adolescence as psychosis. And again, trying to understand how the genetic glitches influence the brain and how the brain results in a particular form of, you know, psychiatric illness, that is not very well understood right now. But the basic sort of architecture of the genetic contribution, the genetic architecture of the disease, starting to reveal itself to us. And that seems to be a common theme among disorders of the brain.